And I will send the recording, a link to the recording out to everybody who was registered. So take it away, Baxter.
Caroline, thank you so much. That was such a great intro. Fair warning, everyone, I may be less articulate now than I was at 18. It's a tough act to follow myself. Let me share my screen. Are you guys seeing the right thing? Are you seeing my slides?
Yes, we can see them.
Okay. Before I dive into everything, just I already thanked Caroline for the intro. Thank you all for coming. It's always flattering to have an audience for anything I talk about related to my research, and also just a huge thanks to CREA in general. Like Caroline said, kind of the starting point for me as both a scientist and an educator. So this is a really cool full circle for me to be able to be back talking to you all. While I'm kind of in the zone with saying thank you, I'm actually going to start my talk with acknowledgements. So the research I'm about to talk about is funded by the National Science Foundation, the U.S. Department of Agriculture, and the U.S. Department of Energy. So anyone who has been lamenting the terrible task of doing your taxes over the past few weeks, maybe will watch my talk and decide that a few pennies of that were the investment. Everything I'm about to talk about was also supported by help of others, namely everyone in the lab I work at here at UVM, the lab of Dr. Stephen Keller, my advisor, who has linked me up with some amazing graduate students, postdoc, lab technicians, and undergraduates to work with. I wouldn't ever be able to do any of this without them. And I've also benefited from a lot of collaborators and friends who have given me advice and data along the way. So now, before I dive into my own research and kind of the details of what you can take away from that research, I find it's helpful to start these talks off by kind of orienting everyone to my broader interests as a scientist. And I'm going to start that off by talking about an experiment, a kind of a famous experiment from back in 1948 that I came across at the beginning of my PhD, and it kind of inspired my work from then on. So this is the experiment of Clausen, Keck, and Hiesi, CKH, we call them, 3 botanists that worked at Stanford University back in the 1930s through the 50s, and they ended up publishing this, like I said, in 1948. So essentially one day, these guys decided to hike up this Nevada range, which you can see in this figure on the slide, all the way up to the highest, some of the highest points in the Sierras, and then back down into the Great Basin Plateau to the east of Sierras. And as they hiked, they collected this plant called mountain yarrow, which is very common in the High Sierra. And they collect samples of the plant, and they collected seeds along the way. And when they got back to Stanford, they planted all of the seeds they collected in something that we call a common garden. So basically just one field where you plant a lot of varieties of the same species. And so while all of these plants were the same species that they collected along this transect, we call it, when you sample in a straight line like that, they identified 2 distinct kind of morphologies, two different sets of these plants, even though they were all the same species. So they found this lowland morph, this taller with more flowers, and they also identified at higher altitudes this dwarf morphology. And what's interesting is that while these plants were shorter, the dwarfs were shorter in the wild, they were also shorter than the lowland plants when they grew them together. So this is a heritable difference between these 2 plants. And so, Clausen, Keck, and Hiesi, they coined this term ecotype that we use to describe when plants of the same species kind of have different characteristics. And they also theorized that these two ecotypes, the dwarf and the regular sized plant, were kind of reflective of the environments that these plants came from. So the idea is the dwarf plant is used to a more challenging and shorter growing season at higher latitudes. So it invests less energy into growth to maintain its fitness at those short growing season, cold locations where it grows. And they describe that process of members of the same species kind of creating these divergent ecotypes in different environments. They describe that process as local adaptation. And in the decades since they published this, local adaptation has become the focus of a lot of research across different branches of biology. And it's become something that I'm fascinated with myself. So to give you kind of another example of kind of the same thing, but shifting focus towards trees now, here's a picture taken by my colleague, Sally Aiken, who's at the University of British Columbia. And so these are Sitka spruce sampled, going from right to left, sampled from Alaska, British Columbia, Oregon, and California. And so we're getting progressively warmer climates as we go right to left. And they're, of course, all planted together. So this is kind of a miniature common garden, like I was just talking about. And this common garden is in Vancouver. So it's, southern British Columbia. And what's interesting is we see kind of the same thing that and Keck and Hiesi saw. We see these two ecotypes. So these tall trees are from warmer environments, and they seem to be growing better in Vancouver. Well, meanwhile, these shorter trees from Alaska and northern British Columbia are much shorter, and so that's reflective. One, that they're probably a bit stressed in the warmer environment of Vancouver, but also these trees from Alaska are used to a very, very short summer, and so one of the hallmarks of this ecotype, this Alaskan ecotype, is that they will enter their dormancy period much earlier in the summer, just simply because they're used to an earlier arrival of winter. So that's an example of the local adaptation that I'm talking about, that forms this short and tall eco. Another interesting thing to take away from this picture is that this tree from California seems to be growing the most, seems to be the healthiest. But like I just said, this picture was taken in Vancouver. And so Sally Aiken, again, who took this picture, she always uses this as an example that, climate change already is shifting environments that trees experience. And it's already creating a bit of a mismatch in the sense that this California tree that's used to warmer environments may actually be thriving in Vancouver a bit better than this British Columbian tree here that's from nearby Vancouver. And so over the past few decades, as climate change has become something that we focus on more as biologists, studying local adaptation has become not only interesting in itself, but it has become a bit more urgent, because now we're not just studying how trees, plants, and animals have adapted to their local environment. We're interested in how adaptations may be disrupted by a changing environment. And so I guess that's an issue for all plants and animals, but we're especially worried about it for long-lived organisms like trees. And that's kind of what this other figure I have on the slide is getting at. This is from a remake of an IPCC figure. And so we have climate projections on the top, and on the bottom is just a reminder that, trees can live 150, 250 years. And so a tree planted not so long ago may experience, something like 3 degrees of warming within its own lifetime. And for a tree, that's significant. So that, in my mind, kind of prioritizes research on these long-lived trees and trying to understand how they've adapted to the environments they're used to and how the environments they're used to may shift in the future. So yeah, I guess kind of repeating myself, in the broadest sense, what I'm interested in with my research is understanding how populations of trees have adapted to their local environment and predicting how those adaptations might be disrupted by a changing climate. So I've mentioned trees a few times, including on the slide. You may be wondering what tree I actually study. I gave it away in the title of my talk, but if you've already forgotten the title, I study poplar trees. Poplar is a term that kind of gets thrown around a lot, sometimes non-biologically. So just so we're all on the same page, when I say poplar, I'm generally referring to members of the Populus genus. And so those are the cottonwoods and the aspens. I focus most of my research on cottonwoods. What is a cottonwood? You may wonder, well, we have two species that grow in Maine that you may have seen. Populus balsamifera, balsam poplar, which I'll talk a lot about today, and Populus deltoides, eastern cottonwood, which I won't talk about as much, but I'll just mention because I bet a lot of you have it in your backyard. Here's a picture of the leaves of Populus deltoides. It makes these long seed pods that have the fluffy cottonous seeds that come out. That's where it gets the name cottonwood. And then Populus balsamifera is similar. The leaves are less heart-shaped and more spear-shaped, and they're also more leathery and smoother. So the range of Populus balsamifera is the tan on this map. And so you can see it's found in Maine, but also all across the northern part of the continent up into Alaska and even the Arctic Circle, which is really cool. Another really interesting thing to me about members of the Populus genus is that they hybridize with each other, meaning they two trees from different species can still cross-pollinate and form fertile offspring. So we'll find poplars that have a parent that's one species of poplar and a different parent that's a different species of poplar. We call those hybrids, which is super interesting. And so you'll notice the range of this balsamifera again here in the tan kind of runs into the range of this other poplar tree, Populus trichocarpa, also known as black cottonwood. So black cottonwood is more common in the Pacific Northwest all the way up British Columbia into Alaska. And Populus balsamifera and Populus trichocarpa hybridize in these locations where their ranges overlap. Super interesting to me. And I bring that up because my talk today will focus, I think, yeah, pretty much 100% on these two species and their hybrids. And so you'll hear me throw around this word takamahawk, which is one of my favorite words I've ever heard. Takamahawk is a word we use to describe these two species a lot of the time, Populus balsamifera and Populus trichocarpa. So a bit more on the takamahawks. They're very shade intolerant and incredibly fast growing when at the sun they require. In fact, widely regarded as some of the fastest growing trees on Earth. They like riparian environments, so along the banks of a river or a lake. And they like it cold. So you'll often find them not only in rivers, but rivers that run through mountain valleys, and they'll grow up the slope of mountains as well. They're known as pioneer species, meaning they're some of the first trees to pop up after some sort of ecological disturbance. So think like a fire, a flood, avalanche, or even the construction of a new road or a railroad. You'll see them a lot of the time. That again owes to their shade intolerance. So they want to be in the direct sun and their rapid growth. So it's easy for them to establish. In addition to that ecological role of being a pioneer species, they also support a lot of other types of life. For example, it's known that over 360 species of moth and butterfly depend on these takamahawk poplars at some point in their life cycle. And of course, there are many other bugs that similarly depend on this tree. So very important. One other thing I wanted to mention, well, actually, first of all, for anyone wondering about these photos, I took all three of these pictures in Alaska. And this one in the bottom right is actually on the roof of the hotel I stayed in. I was there for a conference last summer. And so that's just testament to how well these things perform as colonizers of new environments, because this is a poplar sapling. It's probably 2 years old, growing on the bare roof of a hotel. I think this is like the 18th floor. And then the final thing I want to mention about these takamahawks is they're well known often because of the balsam resin that they produce on their buds in the spring and fall. And actually where the name balsamifera comes from is the balsam, that sap, that aromatic, sticky resin that you find on the buds. Turns out the indigenous peoples of North America, many uses for this sap that I always want to mention, and for other parts of the poplar. So some examples, a lot of First Nations people of Canada and Northern US boil poplar buds and then drink the tea, the result of tea, as a cold remedy. Similarly, the resin used on wounds as a topical treatment. Boiled bark is used as a mouthwash. So like maybe one of the first examples of Listerine was actually poplar based, which is cool. And they had tons of other uses, like for instance, beaver traps. You can use poplar resin as one. I don't recommend it though. I've tried it, kind of messy. And they would even burn poplar bark and feed it to dogs and feed twigs to horses when they were sick. It actually helps reduce things like worms or other parasites that animals can get. Interestingly enough, it's not just beavers, dogs, and horses that enjoy poplar for various reasons. We actually observe bees visiting poplar buds all the time. Even though there's no pollen on the bud, it's a vegetative bud, the bees will collect the sap. This is actually a video. I think I can get it to play. Yeah. So this is a video I took last summer of a bee visiting a poplar bud to collect a little sap for itself, and it flies off. And what it will then go and do is they line the inside of the hive with poplar sap, and it acts as an antifungal treatment. So it helps them defend against fungal infections in the hive. Humans also have, in the modern age, come up with a ton of industrial uses for poplar. Perhaps one of the reasons why it worked so well as medicine for indigenous tribes back in the day is because poplars are actually chock full of this compound called salicylic acid. Salicylic acid is what we discovered aspirin from. Aspirin is derived from salicylic acid, so it has this anti-inflammatory property that is really cool. Poplars are farmed and cut for timber. This is a picture of a 12-year-old poplar stand in an industrial logging operation, I think in Minnesota. And so, that's kind of testament to the growth of these things. These are maybe 30 feet and they're only 12 years old and ready to be logged already. Fortunately, the rapid growth means the wood is not the best quality. So you'll see poplar wood in things like pallets, plywood, store box, chopsticks, kind of like low value timber products. The one exception is skis. So a lot of cross-country skis and downhill skis will have poplar or aspen cores. And those are actually usually laminate. So it's lots of thin layers of poplar wood that are laminated together. Speaking of those laminated products, another one is mass timber. I don't know if people have heard about this, but I always mention it because it's a really cool new use for poplar wood, where they cross laminate. So they switch the orientation of the wood each time they laminate it together. And that's supposedly makes a building material that could one day replace concrete. And in the bottom right here, I have a picture of a building in Wisconsin that was built from this mass timber, as well as conventional boards. Planted windbreaks and agricultural fields, which is another cool usage. They use erosion prevention, and they grow well on the side of rivers, so that's another use for them. They're planted in areas where the soil water is contaminated to bioremediate those areas. And if you remember my first slide, my acknowledgement slide, a lot of work that I do is actually funded by the U.S. Department of Energy, and that is because poplars are seen as a potential alternative energy source, whether it be through burning of biomass or converting them into biofuel, like a biodiesel. And then finally, I always mention kind of beyond industrial applications, there's also kind of artistic applications of poplar. So poplar wood takes paint very, very well. And so a lot of famous paintings are actually painted on poplar. No one ever believes me, but the Mona Lisa is painted on poplar wood, which is really cool. And this is one of my favorite paintings here. It's a Monet. Monet went through a long phase where he was obsessed with poplars and actually painted a ton of them. So they pop up in Monet paintings here and there. My birthday is coming up if anyone likes my talk today and is sitting on a couple extra $1 million, feel free to buy me one of the Monet poplar paintings. Anyway, that's enough rambling about poplars themselves. If people have questions, about any of what I just talked about, we can talk about it at the end, but I should get into my my actual research here. So the basic arc for my research, my approach to the work I do with poplars is very similar to the Claassen-Kecken-Hiesi study I was talking about at the very beginning. So we go out and sample poplars from the wild. And as you could probably tell from those range maps I was showing, poplars grow in all sorts of different environments. And so we try to sample them from as many different environments as we can. Something that's really helpful about poplar is it can be propagated from dormant cuttings. So all we have to do is we go out in the winter, and I hope you can kind of see in this, we've cut, maybe 100 2 to 3 foot long branches. Those get cut into shorter twigs, and we bring those into either a growth chamber or a greenhouse, and we stick them in these things called containers, these little plastic containers that just hold soil. And actually, we don't have to use any nutrients or rooting hormone or anything. Poplars will just start growing roots and leaves in these containers if they're exposed more. Eventually, they end up looking like this. This one actually overgrew to the point that it was coming out the side here. That's why I took a picture of it. But then this is what we will go and plant in common gardens, just like Claassen-Kecken-Hiesi, so we can compare the differences between these different poplar ecotypes. And so finally, after a couple years, we get these trees. And this is, in this picture, my advisor, who for reference is about 6 feet tall, you can barely see him. He's trying to measure this tree. So this is a 2-year-old poplar tree, and it's pretty, I think we measured this one at 5 meters tall, so almost 50 feet. And so that, in a nutshell, is why the government pays us money to study these things, because they just grow so fast, even when they're, in an environment they're not used to. So that first step about is sampling poplars from the wild. I started my PhD in 2018, and before that, my advisor had done a ton of work to collect poplars from all of the locations you see here. And so he and I sat down and we kind of looked at this map and we tried to identify holes. So the two big holes we saw is one, New England. We're based in Vermont, and we only had a few samples from Vermont and none from the rest of New England. And then the other is this area here that I pointed out to you a few slides ago, where balsam poplar and black cottonwood hybridize, kind of in British Columbia, Washington, and Alaska. So in the winter of 2018 and 19, we went out and we filled the first gap in New England. So we sampled poplars growing in the wild all over Vermont and the Green Mountains, all over the Adirondacks, northern New Hampshire, and we even got into western Maine, sampled some beautiful poplars from the Carabasset Valley, Rangeley Lake area, where if you're looking to see balsam poplars in Maine, I recommend driving out to like Kingfield, Carabasset Valley. There's tons of them growing out there. In 2020, we were lucky enough to get a ton of money from the National Foundation to go and fill that other gap. So now we're back. This is Alaska, Yukon, British Columbia. Here's Washington State, to orient everyone geographically. And here we did transect sampling, like I was talking about, Claassen-Kecken-Hiesi, where we basically tried to follow more or less straight lines. You can see we got off track many times. So what's going on with the color on these points? Well, remember I mentioned that in this area in the Pacific Northwest, in the Canadian Rockies, the two species of poplar I focus on hybridize. We did genetic testing to figure out which species each of the sample are. So these bright red points are pure black cottonwood individuals. The darker blue are pure balsam poplar individuals. Anything that is pink or purple is a hybrid. So it has one parent from one species and another parent from another. And I really just point that out because you'll see points colored in this exact same coloring scheme in later slides in my talk. And that's referring to this same thing. So in case anyone was curious, that's what those colors are designating is the ancestry of those samples. So once we had all of those samples, we wanted to plant them into common gardens to compare them. And so here's the location of the 5 original common gardens we planted them into in Alaska, Saskatchewan, Canada, North Dakota, Virginia, and of course, Vermont, where I'm reporting from today. Each one of these gardens has at least 1,500 trees growing in it. Ours in Vermont is over 2,000 at this point. So they're really, really big common gardens filled with as much diversity of poplar ecotypes as we possibly could find. We decided those 5 weren't enough. And so we expanded to these mini common gardens designated by these red points in this picture. So the mini common gardens only have 1 poplars each. So a small subset of all of the samples we have, but we're really excited about these ones. This one here in San Antonio, Texas, in Georgia, another one in Virginia, and one in coastal Maryland. We're excited about those mini gardens because they're kind of locations that are very, as you can imagine, very climatically different than what poplars are used to hailing, from Canada and the northern US. And so our logic there is planting poplars in these locations can give us little hints as to what we might expect the effects of climate change might be, planting them in warmer locations. So we measure many aspects of these trees, and we're constantly observing different traits of the trees. I would never have enough time tonight. I'm already running out of time to talk about all of them. So what I want to focus on is something called phenology. And if phenology is a new word for you, that's kind of a biological term for the stages of the life cycle of a living thing. And so for trees, something that an aspect of phenology that's really important is this dormancy cycle. So following the dormancy cycle clockwise in summer, the tree is obviously actively growing, but then as the days get shorter in the fall, that triggers this process of bud set, where the tree stops growing and forms this big bud at the end of each stem. As the days get even shorter in winter, eventually the tree will lose its leaves, as we're all familiar with, and that bud will fully harden, and all growth stops, and it's prepared for winter. Changes in temperature eventually trigger this event called bud flush. And bud flush is when the new leaves emerge from this formerly dormant bud in the spring. Bud flush is an incredibly important event for the tree, and it's driven, like I just said, by changes in temperature. So thinking about climate change, something that concerns me is that trees are used to a certain change in temperature that indicates spring and a good time to undergo bud flush. And I'm concerned that climate change will shift that signal, and we'll start to see what we call a phenological mismatch, where trees are emerging at the wrong time. So you might be wondering, does that ever happen? Yeah, it does. So we observed this in 2021. In mid-April, we had a really warm week, followed by a snowstorm and a return to freezing temperatures. And so some of the poplars we were growing in our common garden underwent bud flush during that warm week, and then the cold came in. So what happens when these newly formed leaves freeze is that they eventually die. And it's kind of hard to see in this picture, but this is one of the trees that went through that. And you can see this blackening of the leaf. This leaf will eventually turn all the way black and die, as will these other kind of wilted leaves. So properly timing this event, bud flush, is really important for the health of the tree. In fact, it's so important that poplars have kind of evolved this really interesting mechanism of making sure they don't flush too early. And so basically the way this works is there are 2 stages to the dormant period of the life cycle. And remember, the dormant period is when these trees are overwintering, they have no leaves. So it starts in autumn, early winter, the poplars are in this period called endodormancy. During endodormancy, they're basically counting how many cold days they've experienced. And that's a way of making sure that winter really has passed. So once they've hit a certain amount of we call it chilling units, they've chilled enough, that triggers this second kind of checkpoint, which we call eco-dormancy. So they get enough chilling or eco-dormancy, and that's when they start, rather than counting how many cold days they've accumulated, they start counting how many warm days they've accumulated. So again, endodormancy is to winter has passed, and eco-dormancy is a check to make sure spring has progressed far enough. And after enough warming days, that's when the bud break or bud flush takes place. A picture of a poplar that has timed its bud flush perfectly, or at least in Vermont. They've done it right. They flush around the same times the dandelions that you see in the background have bloomed. So when we measure this, we're looking at every single tree in our common garden throughout the spring, and we're noting the date that the leaves emerge from this dormant bud. We see they look like this. But the units that we measure it in, we call cumulative growing degree days. And I bring that up because it kind of confuses people a lot. It confuses me. But what a growing degree day is we're not counting the number of days until the trees flush. We're counting the number of warm days. So if there's a kind of older day, it doesn't count towards that accumulation of warming, because the tree won't count that day. They need warm days, like I was just saying, to break that eco-dormancy. So we measure it. We're trying to use a unit that's similar to how the tree is interpreting the environment around it. So after years of making these observations of bud flush timing across many environments, we've kind of identified this trend that's constantly repeated. And I'm trying to show you this trend in this figure here. It can be, there's a lot going on, so it can be complicated. So I want to work through it carefully. But basically what's going on is we have 3 panels, right? Each panel is a different one of those common gardens I was talking about. So this is North Dakota, ND. Here's Virginia, VA. Here's Vermont, VT. Each point is a tree growing in one of those common gardens. And the color, again, is what I was talking about earlier. So the reds are pure Populus trichocarpa. The blue ones are Populus balsamifera, balsam poplar, and then the hybrids are in purple or pink. On the bottom, on the x-axis, what we're doing is we're showing you information about the home environment of these trees. So not the common, but where we sampled the tree from in the wild. And we're plotting the median number of days below 0 degrees. So basically, a point on the left side, let's look at Virginia. So a point on the far left-hand side is a tree from a very warm environment. Point on the very far right-hand side is a tree from a much colder environment. The final piece here is the y-axis, which is what I was talking about with the cumulative growing degree days. So a point that's very high up on the y-axis, that's a tree that needs many warm days before it will flush its bud in the spring, before those leaves emerge. A point that's very low on that y-axis, it doesn't need as many warm days. It will flush very early in the spring. And so now I'll try to explain this trend that we see. I call it kind of a leaning quadratic trend. Maybe that will mean something to someone. But what we see is that these poplars on the far right, and it's very pronounced in Virginia, that's why I'm kind of focusing on this Virginia panel. These poplars on the right that are from very cold environments are almost always the first to flush. They don't need very many warm days before their leaves emerge. Meanwhile, the trees that do need a long period of warming before they'll flush leaves are trees kind of from these intermediate environments in the middle of the axis. So that's why we get this gray line is kind of trying to show you the overall trend. That's why we get this kind of inverse parabola here, this N-shaped trend where these trees from intermediate environments are delaying bud flush. And what you see with the color is that the trend kind of holds up regardless of if you're a populus trichocarpa tree, a populus balsamifera tree, or if you're a hybrid. So that's interesting that we see this trend across common gardens and across species of poplar. I showed you a bunch of common gardens on that map. We have data for multiple years. And so this slide is just showing you this trend reappear year after year in almost every common garden. The one exception being this panel here. This is Fairbanks, Alaska in 2010, which is one of the coldest winters we've ever experienced in this experiment. And the trees here on the far left, so the trees from the warmest environments, they got super confused. And some of them died. Some of them were just really stressed, and they ended up delaying their bud flush a great deal. So that's the one exception. But this trend even holds up. This figure here on the right is from a completely different research group at the University of British Columbia, looking at a completely different set of poplars in a completely different field. They're even using a different metric. They're asking how cold each day is, not how warm it is, but they still see the same trend as us, where trees from intermediate latitudes or intermediate environments are delaying their bud flush the most. So how do we interpret this? How do we explain this biologically? Well, here's the model that we and other poplar biologists have come to accept over the years. So we think this is the same figure I showed you 2 slides ago, by the way, just here as a visual aid. We think that these poplars that are from intermediate climates, and we've looked at climatic data to support this. I think intermediate climates, so not so warm, not so cold, they actually experience a great deal of variability in spring weather. So there will be the scenario like I was talking about when I showed you those poplars that flushed too early. There'll be these long periods of warm, followed by a period of cold, followed by a period of warm, by a period of cold. It's very confusing in those environments. So in order to just avoid that confusion, these poplars have locally adapted to delay bud flush until later in the spring, when it's certain there won't be a return to any freezing temperature. Meanwhile, poplars from colder climates, which I told you are always the earliest to flush, they require the fewest amount of warm days before they flush their buds. The theory is that poplars from colder climates are usually from high altitude, so high up on mountains, or high latitude, so very northern locations. And so those 2 types of environments, very, very short growing seasons. And so we think these poplars are willing to flush their buds very early in the spring, simply because they're under a lot of pressure to grow as much as they can in a very short season. For them, just of how the summer is at high altitude or high latitude, it's worth it to risk cold injury and just to flush their buds a bit earlier. The final thing I want you all to observe here is that North Dakota on the left and Vermont on the right are both much colder environments than Virginia. And when we look at Virginia, we see as a whole, these are all the same trees in 3 gardens. So the set of trees we have in these 3, as a whole, all of them are flushing much later. They're requiring many more warm days in this warm Virginia environment than they would in the colder Vermont or North Dakota environments. And so we basically interpret that as reflecting that endodormancy period I was talking about. So they require a number of cold days before they're ready to start counting warm days. And in Virginia, the winter is just so mild, they actually never get those cold days. And so in the days of spring, still trying to count cold days that never come. So they actually need to use the colder nights, the colder evenings of spring as a surrogate for that, and that just causes a delay in their bud flush. I'm almost done here. I'm on my second to last slide. But I mentioned earlier, we were really excited about those common gardens in Georgia, Texas, Maryland, and our additional one in Virginia, because I was saying those are much, much warmer environments than poplars ever experience. And so what we see in those common gardens is kind of, I guess here on the right-hand side, this is Maryland and this is, we see kind of the same trend that we're used to seeing, where trees from colder environments are flushing the earliest. But then when we get into these really warm environments, Georgia and Texas, they're nothing like these poplars have ever seen in their lives. Basically, everyone delays bud flush a great deal, to the point where in Texas, we actually see a reversal of the trend we're used to seeing. This N shape has turned into a U shape. And so these two poplars here, these blue points, they're from Alaska, one of the coldest environments. That's why they're so far to the right. They just have no idea what's going on in Texas. They're waiting for a nice cold winter that, like I was saying, never arrives. So they're still waiting. They arrive in mid-April in Texas, and they're still waiting for cold weather. And eventually they will just flush their bud, more or less because they have no option other than to go through with it. But they're just simply confused. So from all of this, all of these observations at different gardens at different years, we've kind of come up with two main concerns for poplars looking at climate change. And so first one is for poplars in the coldest environments, the highest altitudes or the furthest north. We worry that, like I was saying in the previous slides, those are always the trees that require the fewest number of warm days before they flush their buds. And so we worry that in these cold environments, it may become more common to have late winter warming events followed by freezing temperatures. So the weather that I said was more common in intermediate environments, not in very cold environments. With climate change, that may happen more in these colder environments. And we're worried that poplars from those environments are not used to that. And so they'll get tricked, so to speak, into flushing too early and then getting frozen, like in this picture here. So in order to figure out how big a deal that will be, the next step in my research is looking at these poplars from very cold environments and testing their tolerance to these early spring freezing events. On the flip side, we're also worried about the poplars from some of the warmest environments, because we're worried those will be the first environments under climate change to start looking more like Georgia and Texas, where, remember, we saw basically all poplars severely delaying their bud flush, requiring many, many warm days before they would flush their buds. The thing is, like I mentioned, poplars being a riparian species, they expect there to be a great deal of water around their roots when they start growing leaves. And so the concern in these warm environments is that in climate change, they may start flushing their buds well after spring rains have dried up, which is not ideal for the poplar. So to figure out how big a deal that will be, we're also looking closely at these poplars from very warm environments, and we're testing their tolerance to drought in the early summer. And I will stop there for questions, but kind of like I did back when I was 18, talking at the annual meeting, I will try to leave us on kind of an optimistic note, which is that I have started experiments testing both of these things. That's what both of these pictures are for. And let me tell you, these two poplars, yeah, they had flushed their bud way too early and got frozen, but they did bounce back. And I'm doing more tests on that now and seeing incredible resilience to cold injury. These two poplars, every single leaf ended up dying. This is a drought experimentally imposed drought experiment we did, and it caused every single leaf to die. And I was about to throw these 2 away. I had them set aside, forgot about them, and they came back from their roots. So, you know, climate change is going to throw a lot at poplars and, you know, every organism on Earth. Eventually more than they can handle, but we're learning that they can certainly handle a lot. At this point, I'll gladly take questions. I'm just going to leave it on a slide that shows 4 pictures. This is our common garden in Vermont. And so the 2 pictures on the top are the before, and the pictures on the bottom are the after. Both of these on the bottom, actually this one on bottom left is taken from the exact same spot as the picture on the top 2 years later. So these are 2-year-old poplars compared to the day they were planted. And yeah, like I said, I actually will stop now, and I actually will drink some water and take some questions.
Wow, that's amazing. I had no idea that poplars grew that fast. That is quite something. So folks are welcome to either unmute themselves and ask a question, or pop a question into the chat, and I'll funnel them to Baxter while we're waiting for that. I was curious, Baxter, what does the term Takamahawks come from?
Oh my God, I'm so glad you asked. I'm so glad you asked. I never have time to cover it, but I always mention it in the hopes that someone asks. So I have a side interest in linguistics, and the origin of Takamahawk has this crazy linguistic history where it's actually, it's borrowed from the Mayans, who never saw a poplar probably in their entire life. But the Mayan people of, you know, Mexico would extract plant resins from a variety of plants and use them for medicinal purposes. And their word for any medicine derived from a plant resin was something similar to Takamahawk. The Spanish, who eventually you know, conquered the Mayans, they stole that word and incorporated it into Spanish, which is where we get Takamahaca. They added an A at the end, I believe. That somehow got super confused by the British, who for a while used it again to mean any resin-based medicine. But in the colonies of what would become America, basically the only plant resin-based medicine was from poplars. So it slowly came to just mean poplars that produce resin, aka the Takamahawks. However, you see it getting misused to this day in botanical textbooks and stuff, because it turns out the Algonquin peoples, including the Abenaki that you know, live in Maine and northern New England, they had a very similar word, tamarack or something like that, that meant larch, right? A completely different type of tree. And that's where we get the term tamarack. So all over the place in early botanical literature, the word Takamahawk and tamarack get mixed up, even though one is from the Mayans and one is from the Algonquins, thousands of miles away, talking about different trees for the same purpose. Hopefully that was more of an answer than you were expecting.
I just love the fact that you drilled down that deep to learn where that word came from. So we do have a bunch of questions, so I'm going to start working through those. Let's see. Do you have any sense of the biological mechanism the poplars are using to count cold and warm days? Oh, what a good question.
Great question. Great question. Another one that I hope people ask, because I don't have time to go into it that much. So there's a lot of biological mechanisms all going on all at once. And so I won't be able to describe all of them, and we actually don't know all of them, and we're still studying, but a lot of it comes down to genetics, the expression of specific genes at specific times throughout the winter. But one of the major drivers is super interesting. So plant cells are connected to each other with pores called plasmodesmata. You might remember that word from biology in high school. So plasmodesmata are just these little pores between the cells that signaling molecules can get passed through. During dormancy in the winter, poplars will fill up those plasmodesmata with something called callus, which is like this fatty compound. And that's to make sure no signals get passed through to repress growth in the winter. And so one of the ways that they're counting the amount of cold is actually the process of that callus freezing and dehydrating. And so the thicker, more expanded that callus is, and the more dehydrated that the tree finds itself, that's a pretty good indicator of how cold it's been. Then, in order to count warm days, that callus starts to melt. And one of the only signaling molecules that the tree is expressing during dormancy is this thing called fatty acid desaturase. And that is a compound that can break down the callus. So as it gets warmer, that it's an enzyme. Enzymes work better when it's warmer. And so as warm days accumulate, this fatty acid desaturase is breaking down the barriers in the plasmodesmata faster. And eventually it digests the whole callus and unclogs the plasmodesmata. And then the signal to start undergoing bud flush, it basically has to travel from the leaf into the bud, and it can't, from the stem into the bud, and it can't make that journey when the callus is clogging these pores, this plasmodesmata. So once the callus is out of there, that allows the signal to travel to the bud, and that's what triggers bud flush. So that's the major driver. But again, like I said, there's also a lot of other things going on.
Wow, you're very good at explaining complicated things to us lay people. All right, another question. How many trees are in the mini gardens?
So the mini gardens, the direct answer to the question is 100-ish. Of course, they don't always all survive. So it's usually like 80 to 90. And it's 2 replicates of 50 trees. We went back and forth about whether we just wanted to try one shot with 100 different samples. But we ended up kind of hedging our bet by we planted 2 replicates of 50 samples.
And are the twigs that you're starting from, do they all have to come from the end of a branch, or could you cut one branch into multiple pieces and turn all of those into grow all of those separately? Yeah,
for big mature trees, which is usually what we try to sample from, so we're not, hurting the tree very much. We'll cut one big branch, and then we'll take that branch and we'll cut off all of the like 1 year growth, we call it. So one branch at times will give us, like 20 to 30 twigs that could all be propagated.
Avril wants to know, how long will this study of poplars in the gardens go on?
Well, the short answer is for as long as we can convince funding agencies to keep giving us money. So I think the pictures I'm showing right now, this garden, the funding for half of it extends through next summer, and then it dries up. So at that point, we can usually keep it going for another year. And then what happens is we call it coppicing. So we'll go through and we'll cut down every single poplar. Poplars are crazy though, like I already mentioned. So when you cut them down, it does not kill them. They will come back from the stump or from the roots, and they'll usually send out 2 or 3 shoots. So then we actually get bigger trees after we do that. So we'll usually, the typical cycle is you'll coppice your trees after about 4 or 5 years, and then try to get funding to keep going when they regrow. So you like have an off year to go apply for grants to keep going. Yeah, so the other half of the gardens I'm showing were funded actually by the University of Vermont, more or less into perpetuity. So at the very least, we'll still have half of that big Vermont garden forever. And hopefully we can find funding to keep all the other ones going as well.
Interestingly, Nick wants to know, how do you protect the seedling trees from being munched by deer, and how long is that protection necessary?
Deer have become the bane of my existence, and more so bane of the poplar's existence. So this garden I'm showing you again in these figures at the University of Vermont Horticultural Center. That whole horticultural center is surrounded by a massive chain link fence, and they put it up in the 80s to keep deer out, because it's not just poplars we grow there. There's a ton of crops that get grown, and obviously deer are an issue for all of them. However, when they put the fence up, they actually trapped a family of deer inside the horticultural center, and we have not been able to get them out. So we've tried a number of things. We've tried fox urine. We've tried different soaps supposedly scare deer away. The one thing we've done that works pretty well is actually, you can't see it in these pictures, I don't think, but this entire garden is now surrounded by a fishing line fence. So we do wooden posts with 3 layers of fishing line. And I guess the deer hit it and they don't like it very much. Ever since we put that up, we'll have like 1 or 2 poplars munched on by deer per winter. Meanwhile, our collaborators in North Dakota didn't do that. In their first year, I'm not kidding, I think they lost about 80% of their garden to deer. And that is an issue throughout the life cycle of the poplar. The deer, they like that apical bud, the very top bud, so they do go for the shorter ones. But I mean, they can reach pretty high up, so they will keep coming back year after year. So we have to keep that fishing line up.
So when you say, I'm curious about this fishing line, because I'm sure many people here have the same problem with deer in their yards. Are you saying like it's a fence made out of just 3 horizontal strands of fishing line, and they run into one of them and it deters them?
Yeah, something about it. I'm not sure what, I mean, deer are so weird. I think it's being touched by something they can't see is weird to them, so they run away.
All right, I'm gonna try that. Are there many or any Takamahawks in the Cadence River Preserve? And if so, where would they be?
I would love to find a balsam poplar. So yeah, if there was a Takamahawk in the Cadence Preserve, it would be a balsam poplar. It would be kind of rare finding one that far east in Maine. Like maybe you saw on my sampling map, the ones we sampled from Maine, from very far western Maine. That said, my mom lives in Brunswick. She has one growing in her driveway. Not 100% sure that's not a seed that hitchhiked on me somehow, because I'm constantly touching poplar seeds, but I like to believe it's native, and so they can grow that far east. I've seen them growing along the Androscoggin as far south and east as Lewiston. So it's totally possible. And if you wanted to find one, I would say along the bank of the river, probably as close to the water as you can get. That's what they like.
Okay. Sarah asks, what's the difference between poplars and aspens?
So there's, not very many differences, because they're very, very closely related. I always refer to them as cousins. So the aspens, Aspens are also very fast growing. They're also pioneer species. A major difference is aspens will flower and put out pollen much earlier than poplars. So that's one of the reasons they don't hybridize as much as the cottonwoods do with each other. And aspens are better adapted to go farther away from water. So that's why you'll see aspens, especially in the West, growing way up into the mountains up until you get to be like pure conifers. They're a bit better adapted to those rocky outcrops and serpentine soils like that. That's probably the only real difference, though. It's really easy to confuse aspens for poplars and poplars for aspens. And in captivity, so to speak, they do cross-pollinate. They just don't as much in nature.
So Jim says, trees can count. Cognitive capability, please elaborate. I think you sort of answered that, but Jim, if he did not, if he did not answer that, I mean, is there any more to your answer? I feel like you sort of answered that with the explanation that you gave earlier.
Yeah, I mean, I'll just reiterate again that it's a pretty cryptic process. I mean, we understand the counting partially comes from the callus clogging the plasmodesmata, but there's a lot that we don't understand about it. And, I'm willing to think of it as some sort of intelligence. I mean, I certainly can't keep track of the weather for that long. So however they're doing it, yeah, I think it's fair that there's some very basic level of cognition. I'm sure my friends in neuroscience would not agree with me at all for a second, but as a plant biologist, yeah, that's the closest thing to cognition I'm ever going to experience. So we can call it that.
Well, and there's also all that's going on in the underground, communication that's taking place, and above ground, communication between trees that sure seems to resemble a something akin to a nervous system. But anyway, I think if anyone, I think I got all of the questions, so that might, I'm just checking. I think that is it, but that was incredibly, that was totally delightful, Baxter. I learned a ton, and I'm sure everyone else did. It sounds like great research. And what's next for you?
Yeah, kind of like what I was hinting at in that last slide. I'm moving into doing a lot of research on cold tolerance, especially tolerance to freezing events that happen right after poplars undergo bud flush. Again, kind of out of concern that climate change will make spring weather more variable, and trees from northern locations will potentially get tricked into flushing their bud before winter is all the way over. So I've been doing all these. Actually, haven't been outside as much as I usually am in the late winter and early spring. I had one picture in my slides, but at UVM, we're lucky enough to have these amazing growth chambers that we use, which is essentially like massive refrigerator with lights in it, where we can control every aspect of the environment, especially the temperature. And these growth chambers can go down as cold as 14 degrees Fahrenheit. And so what I'm doing is I have two growth chambers. One is at like a nice balmy 55 degrees, and I'll put dormant poplar cuttings in there. I'll wait until the moment they undergo bud flush, and then I'll grab them out and I move them to a different growth chamber that's at 14 degrees Fahrenheit, and I see if they can survive that. So pretty basic experiment. We have another thing going on right now. It's called an electrolyte leakage assay. So we basically take a hole puncher, believe it or not, and we take a bunch of punches from a poplar leaf, and then we put them in basically a chemical solution that conducts or that doesn't conduct any electricity, expose them to a very cold temperature. And if that cold temperature has damaged the leaf, those little hole punches of the leaf, they will start to leak electrolytes because their cells have popped due to the cold temperature. And so we can then measure how much electricity is conducted through that liquid that originally didn't conduct electricity. And that's a metric for how much damage has happened on the leaf. And so as you can imagine, we can do thousands of those measurements at once. So I've been trying to do it at really high input, getting better at it slowly. Yeah, that's basically what I'm up to now.
Well, I think you have a brilliant future as a teacher because you're very good at explaining complicated things to us lay people. Well, thank you so much, Baxter. And this was wonderful. And I hope that maybe there's someone in the audience who's going to ship a nice Monet off to Poplar. I wish it could be. I wish we could compensate you in that way, but I don't think that's in our budget. But there's lots of kudos in the chat. But thanks so much for taking the time to share your research with us. This was really enlightening. So
yeah, thank you so much.
Thanks everyone for showing up, and I'll send out a link to the recording if you want to share it with others in the next couple of days. So good night, everyone.
Night, Bex.